Camphor. Introduction. Why did you choose this question? To find the most effective antibacterial agent against this potentially deadly bacteria. What's our previous knowledge? Some strains of E. coli cause food poisoning. This bacteria is in the intestines of warm-blooded organisms. Important functions: the production of the vitamin K2, the prevention of other harmful bacteria from entering the intestines. Hypothesis: the most effective disinfectant will create the largest zone of inhibition because it will inhibit the growth and reproduction of E. coli.
Method and materials. Variables. The independent variable in this experiment was the type of disinfectant used, and the ones we used were Purell, iodine mouthwash, and bleach. And the dependent variable in this experiment was the size of the zone of inhibition, which we measured in millimeters. And the control variables were the temperature of the room, the humidity of the room, and the air pressure.
Methods and materials, control and experimental groups. The control group. Each quadrant is compared to itself. There is no direct control group. Experimental group. Each disinfectant would be considered an experimental group. How the control and experimental groups differ. The experimental and control group had no distinguishable difference. Data collection. Record the diameter of the zone of inhibition of each disinfectant in every quadrant. Methods, materials, procedures. There were 7 of those. And in our data, the data shows the relationship between the variables of what is the most effective disinfectant created the largest, and the most effective disinfectant created the largest zone of inhibition. Discussion, sources of error and limitations. Sources of error include possibility of contamination, weak E. coli culture, incubator temperature, and then our techniques used could affect it, including inoculation techniques, sterilization procedures. And there are some limitations.
Discussion improvements to the experiment and further research. There are a few improvements that can be made to this experiment, which include using larger sample sizes and repeating the experiment. We can also use more E. coli cultures and use stronger ones in it. We could also use a more equipped lab and better materials. And for further research, we could look into how each antibacterial substance works. Conclusion. The hypothesis was proven correct when the most effective disinfecting agent created the largest zone of inhibition, which was iodine.
Thank you very much. So at the end, that's the end of our presentation.
That's the end of our presentation.